Gel electrophoresis separates DNA. We load samples into an agarose gel. An electric current pushes them through. Smaller fragments move the fastest.
Because DNA has a negatively charged phosphate backbone, it moves through the gel toward the positive electrode when an electric field is applied.
First, we prepare an agarose solution by dissolving agarose powder in an appropriate electrophoresis buffer, such as TAE or TBE. The mixture is heated carefully until the agarose completely dissolves. This produces a clear solution. After heating, the molten agarose is allowed to cool before casting the gel. It should be warm and fluid, but not excessively hot. Now we prepare the casting tray and position a comb. The teeth of the comb will create wells in the gel. The molten agarose is carefully poured into the casting tray, forming a uniform layer. Care should be taken that there are no air bubbles. As the agarose cools and solidifies, the comb is removed, leaving small wells where the DNA samples will be loaded. The solidified gel is now placed carefully inside the electrophoresis chamber. Running buffer is added until the gel is covered. The buffer provides ions that allow electric current to pass through the system. An important point is orientation. The wells are positioned near the negative electrode, because negatively charged DNA must migrate toward the positive electrode. Finally, the electrodes are connected to the power supply, but the power remains switched off while we prepare our DNA samples.
Now we prepare the DNA samples that we want to separate and analyze.
Before loading DNA, we mix the samples with loading dye. The dye helps the sample sink into the wells and allows us to monitor its movement. Using a micropipette, the required amount of loading dye is added to the DNA sample. The DNA and loading dye are mixed gently to obtain a homogeneous sample ready for loading. Next, we use a micropipette with a fresh, sterile tip to transfer each DNA sample into the wells. The pipette tip is carefully positioned just inside the well, without touching or damaging the bottom of the gel. Now the first DNA sample is slowly dispensed into the well. The same procedure is repeated carefully for each sample, using a fresh tip whenever required to prevent cross-contamination. A DNA ladder is also loaded into a separate well. It contains DNA fragments of known sizes and serves as a reference for estimating the sizes of unknown fragments. Once all samples are loaded, the chamber is closed securely. Before applying the electric field, now the power supply is switched on, creating an electric field across the agarose gel.
DNA fragments begin migrating through the agarose matrix toward the positive electrode.
The agarose acts like a molecular sieve. Smaller DNA fragments move more easily through its pores and therefore migrate farther than larger fragments. As electrophoresis continues, DNA fragments of different sizes separate into distinct bands. The run is continued for an appropriate period, depending on the gel concentration, voltage, and size range of the DNA fragments. Once adequate separation has been achieved, the power supply is switched off before the gel is removed. The gel is now ready for visualization, so that the separated DNA fragments can be observed. DNA bands can be visualized using an appropriate fluorescent DNA stain and a suitable blue light or UV-based imaging system. Each visible band represents a population of DNA molecules of approximately the same length. By comparing the sample bands with the DNA ladder, we can estimate the approximate sizes of the DNA fragments. Fragments that have migrated a shorter distance are generally larger, whereas fragments that have migrated farther are generally smaller. If a particular DNA fragment is required for further experiments, its corresponding band can be selected from the gel. The selected band is carefully excised from the Agarose gel using clean laboratory equipment.
The gel slice containing the DNA fragment is transferred into a tube for DNA recovery.
In gel extraction, the DNA is released from the agarose and purified using an appropriate DNA recovery method.
So, gel electrophoresis involves preparing an agarose gel, loading DNA samples, applying an electric field, separating DNA fragments, and finally visualizing the resulting bands.
Gel electrophoresis therefore transforms an invisible mixture of DNA fragments into separated bands that we can visualize, compare, analyze, and when required, recover for further experiments.